Hi everyone, this is Miss Lambert. Today we're going to talk about the four different types of macromolecules, including the structure of those macromolecules and how the structure determines their function and their properties in living systems. If you want to follow along, I have posted these sketch notes on Canvas if you want to print them out and follow along with us. So, when you're thinking about macromolecules, I want you to imagine the macromolecule cheeseburger, because in that cheeseburger, you're going to find all four types of macromolecules. You'll find carbohydrates in the bun, you'll find proteins in the burger patty, you'll find fats in the cheese, and you should find nucleic acids in any vegetables that you might add to it. So, this is the image I want you to conjure when you're thinking about the four different macromolecules. So, the first macromolecule that I want to talk about today are our lipids. Now, structurally, lipids follow this general path or this general guideline of having some type of head with fatty acid tails attached to it. In this case, this is what we call a triglyceride because it has a glycerol head and three or tri fatty acid tails. Now, if we look at an actual triglyceride molecule, we will see that these fatty acid tails are full of carbon to carbon bonds. And lots of bonds mean that if you were to break a fat down, you would release a lot of energy. But right now, in its current state, all that energy is stored away in those bonds. And so that means that the primary function of lipids is to be long-term energy storage in most living things. Now, it's visible on people when we have lots of this long-term energy stored away in the form of fat deposits that we see on our bodies. But as far as lipids in things like bacteria and plants, you don't look outside and see a fat tree or a bacteria with a big belly. They use lipids for other things. And that is where phospholipids come into play. Now, hopefully you remember from ninth grade biology that phospholipids are the lipid that makes up the cell membrane. And the reason that they can do that is because lipids are non-polar. They do not mix with water. We call that being hydrophobic. And so lipids include things like fats, they include things like oil. That's why oil does not mix with water. They include waxes and also steroids like testosterone and estrogen and cholesterol. They do not mix with water because they are non-polar. And so, because they don't mix with water, lipids are a really good barrier between two areas that might be full of water, like the outside and the inside of a cell. But if you were actually zoom in on a phospholipid, you would find that the head is actually polar and the tails are nonpolar. And so, because of this, phospholipids will arrange in a bilayer. So, if we can imagine this side is outside of the cell or extracellular, and this side is inside the cell or intracellular, you can see that these polar heads are going to be close to the water because they're polar like water, and so they um, are hydrophilic, whereas these non-polar lipid tails are going to stick together on the inside. And so the big takeaway from this is that lipids are great for long-term energy because they have lots and lots of bonds, but also that the way they behave is in part due to them being non-polar and not mixing well with water. Now, there are actually different types of lipids. Obviously, I just talked about triglycerides, which is the primary fat that gets stored in our adipose tissues in our bodies. But we also consume lipids. And one type of lipid that we consume are saturated fats. This is the one you hear about on the news all the time that's, really bad for you, supposedly. Um, there's not a lot of evidence that there is such a thing as a bad or good fat, but that's often what they're called because they are more likely to clog your arteries. But a solid or saturated fat is a type of fat where you have all of these carbons bonded to hydrogens. If we were to zoom in to this particular fatty acid tail on this triglyceride, we would see that every single carbon has hydrogens bonded to it. It is saturated with hydrogens. That's why it's called a saturated fat. And the reason that these are quote unquote bad for you is because saturated fats easily stack on top of each other. And so they're more likely to clog your arteries than maybe an unsaturated fat, which is the next type we're going to talk about. These are liquid fats. Triglycerides are considered unsaturated fats. And so we're talking about vegetable oil, olive oil, those things that are considered quote unquote good fats. And this happens when you don't have as much hydrogen inside of the tails as you would normally, because one of the hydrogens has to go away because there might be a double bond or something in the tail structure. And then finally, they talk about the really good fats for you, what you might find in fish. We can have something called polyunsaturated fats. And these are fats that have multiple double bonds in them. And you can see this picture right here. There's not nearly as many hydrogens in this polyunsaturated fat as there would be in a saturated fat. So takeaway is that lipids are nonpolar, which means that they're really good for being membranes and barriers inside of cells, but they're also really great for energy storage because they have tons and tons of bonds. The next one I want to talk about are carbohydrates. So carbohydrates, as the name suggests, are made up of carbons, hydrogens, and oxygens. And this is the structure for all carbohydrates. It's very consistently carbons, hydrogens, and oxygens. And specifically, you can find them in a ratio of 1 carbon for every 2 hydrogen for every 1 oxygen. So you guys may be familiar with this particular formula, C6H12O6. That is what you've been taught that the chemical formula for glucose is. It follows the 1 to 2 to 1 ratio perfectly. But something else to know is that this is also the chemical formula for fructose as well as galactose. So C6H12O6 does not just mean it's glucose anymore, although most times it is. So if we were to actually see this molecule drawn out, this is glucose. It is a ring shape, and you can count the 6 carbons. So this is a carbon, that is a carbon right there, 3, 4, 5, 6. We can also count the 6 oxygens, 1, 2, 3, 4, 5, 6. And there are actually 12 hydrogens, although they don't show all of them here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and there's supposed to be one right here for 12. So something to know is that this is glucose, but I just told you that fructose and galactose also follow this chemical formula. What's different about fructose and galactose is that they are arranged in a different kind of ring shape. So just be on the lookout for those types of differences. And so because glucose is made up of just this one ring shape, we call it a monosaccharide, mono meaning one. But a lot of times, those types of simple sugars are not going to be enough, that you might have to combine sugars into what we call a disaccharide. And so you're probably actually more familiar with disaccharides because this is what sugar is. This is what table sugar is. It's a combination of 2 monosaccharides put together. Now, if you guys will remember from our previous class, the only way you can put these 2 things together is through one of those condensation reactions, and that helps to turn monosaccharides into disaccharides. Now, we don't just get sucrose. That's not our only source of carbohydrate. We actually get most of our carbohydrates from starches and cellulose, from the foods that we eat. And so that's where we get into polysaccharides, where you have lots of these rings put together. If you are a plant eater, which I hope most of you eat plants every day, then you consume a lot of starches. You also consume cellulose. And if you consume too much of that for your body to be able to use in a day, it will get converted into glycogen in your liver. And so you can see how these are polysaccharides. You've got all these rings stuck together. Now, cellulose, starch, and glycogen are actually all made up of glucoses put together. But what you'll notice is that the glucoses are stuck together in slightly different ways for each of these three polysaccharides. Now, as far as why carbohydrates are so important, they, as you can immediately tell, don't have nearly as many bonds as lipids do, but those bonds are easily broken and ready to be broken by your body. And so that makes carbohydrates ideal as a short-term energy source. That is why glucose is the first thing that's broken down when a cell is doing cellular respiration, because it's quick and easy for a cell to break down. Fats make it a little bit harder, and they add a few more steps if you want to start breaking them down. And they're essentially the energy source for all living things. Even if you're a bacteria, you're going to use glucose to do cellular respiration and make energy. We use it to do cellular respiration and make energy. So carbs are essential for our everyday energy needs. The next type of macromolecule that I want to talk about are our proteins. So another term for proteins are polypeptides, poly meaning many, peptide referring to the type of bond that puts these amino acids together. Amino acids are the building blocks of proteins, and they're called amino acids because they are composed of an amine group, which is where you find the nitrogen, and a carboxylic acid group, which is where you get the acid part of amino acid. And when you want to put amino acids together, you link them with peptide bonds. Once again, you can build peptide bonds by doing that condensation reaction that we talked about yesterday. And then you can also break peptide bonds if you do a hydrolysis reaction. The other part of an amino acid is the R group. Now, the R group stands for any of 21 different functional groups that you can tack onto an amino acid. You can have functional groups that are polar, which means they like to hang out with water. You can also have functional groups that are nonpolar, which means they don't like to hang out with water. You can have some that are acidic and some that are basic. And so depending on whatever R group is attached to your amino acid, that's going to determine how that amino acid behaves when it is assembled into a protein. So when we're talking about putting amino acids together to build a protein, that's what we call primary structure. So just what amino acids are you putting together? Are you putting together a serine, methionine, and a histidine? Or are you putting together a glycine, a proline, and a lysine? It just depends on what sequence of these different amino acids you put together. Now, like I said, this R group is going to determine how these amino acids behave with each other. And that is where we start to get into something called secondary structure, and that your protein might start to twist on itself. We call that an alpha helix, or it might start to fold on itself. We call that a beta sheet. And the more that it twists and the more that it sheets on itself, it's going to start to form a tertiary structure where it's going to twist and combine even more. Proteins are only able to carry out their different functions because of the shapes that they take on. And the shapes that they take on are ultimately determined by what amino acids you put together, how they start to fold and twist on each other, and what shape that makes overall. And in some cases, you actually need to combine multiple proteins to make a functioning enzyme or a functioning antibody. And so sometimes you get into what is called quaternary structure, where you have multiple protein pieces that come together. Now, I just kind of mentioned it, but twisting and folding is what gives a protein its shape, and its shape determines how it can act. But sometimes you can cause a protein to unwind and unfold and lose its shape, and we call that denaturing. This is what happens when you cook an egg. The egg proteins exist in one form when it's raw, And then when you heat it up enough, you untwist and unfold the proteins in that egg enough that it becomes denatured and that the protein doesn't exist in the same way anymore. A lot of times, denaturing is irreversible. That's why you can never turn a cooked egg back into a raw egg. And that's why you also have to be really careful if you ever get a fever. If your fever stays above a certain temperature for a long time, you can start to denature the proteins in your brain irreversibly, which means that your brain may not function properly anymore. And so denaturing can happen if something gets too hot. That can affect folding and twisting of proteins, but also the environment. Maybe you're in too acidic of an environment, too basic of an environment. A lot of things can affect how proteins fold on each other. And so that means that proteins are the heavy hitters of the macromolecules. They are the working moms. They have tons of jobs that they have to do. Proteins are involved in structural components. We see that in our muscle cells. They're involved in immunity. All these antibody things that people are talking about with COVID-19, antibodies are ultimately proteins. Transport, they're involved in moving things across the cell membrane. They're involved in cell signaling, and probably the single most important thing is that they are involved in chemical reactions in the form of enzymes. And we're going to talk a lot more about enzymes later on in this course. The last and final macromolecule that I want to talk about today are our nucleic acids. Now, these guys have one job and one job only, and that is to ultimately hold genetic information. And the way nucleic acids do that is that they're made up of individual pieces called nucleotides. Now, a nucleotide is three parts. You have a phosphate group, some type of 5-carbon sugar, and a nitrogenous base. If we were to actually look at the chemical structure of it, we can see the phosphate in the phosphate group. We can see that this sugar has the same or a similar ring shape as our other carbohydrates. And then the nitrogenous base has a bunch of nitrogens in it. And depending on if we're talking about RNA or DNA, the sugar may be different and you may have some different nitrogenous bases. And so these nitrogenous bases are what we refer to as like A's, C's, T's, and G's. And that's actually the code that we talk about in your DNA and RNA. And so the sequence of these particular nitrogenous bases, whether you have AAG or CAG, that's going to determine what proteins are made. And as we just said, proteins do all the things inside of a cell. So it's really important for these nucleic acids to be able to code for the right proteins at the right times. Now, you are already probably familiar with DNA and RNA. DNA is double-stranded. It's in this double helix shape. RNA is usually single-stranded. We'll talk more later in this course about instances when it is not single-stranded. But their main job is to store genetic information. And that, again, comes from the fact that they are made up of these nucleotides that have different nitrogenous bases that kind of serve as codes for how to make proteins.